Okay, so welcome to this next video in which we are discussing the palmitylation of proteins. Okay, so we've discussed that there are two major forms of palmitylation. There is s-palmitoylation and there is n-palmitoylation. And we are going to focus our attention on s-palmitoylation, which is when the palmitoyl group is added onto the thiol group of a cysteine residue that is within the middle of a polypeptide. Okay, now, when you add a palmitoyl group onto a cysteine residue in the middle of a polypeptide, there are two possibilities. Either the palmitoyl group forms what is known as a stable palmitylation, okay, which means that once that palmitoyl group has been put on, it's never going to be removed. Or what can happen is you can get cycling. Okay, so the alternative is that the cysteine residue can continuously have palmitoyl groups put onto it, and then they can be taken off. Okay, and then they can be put back on and taken back off. Okay, so when we're talking about any specific cysteine on a polypeptide which can be palmitoylated, either it will be a stable palmitylation, which means that the palmitoyl group once on will stay on for the rest of that protein's life, or it can be a cycling palmitylation where the palmitylation is essentially reversible and it's continuously being removed and put back on. And of course, when we're talking about these proteins that are being anchored in the inner leaflet of the phospholipid bilayer by their palmitoyl groups, some of these palmitoyl groups will be stable palmitoyl groups, okay, and that will mean that the protein will always be anchored in the membrane. But what's more interesting is when the palmitoyl groups are all cycling, okay, because then the palmitylation is reversible, and hence you can disanchor the protein from the inner leaflet of the phospholipid bilayer by breaking the thioester links between the palmitic acid molecules and the cysteine residues within the protein. Okay, right, so now what we want to study is the enzymes which can put palmitoyl groups onto thiol groups, i.e. the enzyme that is going to take palmitoyl coenzyme A and attach the palmitoyl group onto the thiol group of cysteine residues within polypeptides to form these thioester links. Okay, right, so these enzymes are in the class of enzymes known as protein acyl transferases. Okay, so all of these enzymes will be in the class of protein acyl transferases. Okay, which are enzymes which will add acyl groups onto proteins. Now specifically we are talking about adding palmitoyl groups onto proteins rather than just any old acyl groups. So specifically we are talking about palmitoyl transferases. So protein acyl transferase is just any enzyme which transfers an acyl group onto a protein. Okay, now palmitoyl groups are acyl groups, but there are other acyl groups. For instance, myristor is also an acyl group which can be added onto proteins. So specifically, the type of enzymes we're looking at will be palmitoyl transferases. Okay, now the enzymes which add palmitoyl groups onto the sulfur atoms of cysteine residues, i.e. which are going to take part in s-palmitylation, are a family of enzymes known as the DHHC protein and acyl transferases, okay? And protein acyl transferases, by the way, would often be abbreviated to PATs for short, so P for protein, A for acyl, and T for transferases. So we're going to look at the DHHC PAT family. Okay, right. So this contains a huge number of different enzymes which are all capable of adding palmitoyl groups onto cysteine residues, i.e. they're all capable of catalyzing s-palmitoylation. Okay, so these are going to catalyze s-palmitoylation. Now, there are 23 of these enzymes that are known to exist in the human genome, basically. And confusingly, or annoyingly, a mild annoyance, basically, is that they have been numbered DHHC number 1, DHHC number 2, DHHC number 3, then you go all the way up, and it's perfectly sensible, all the way up to DHHC 9, and I will explain to you why they're called DHHC enzymes, but we'll come to that. Then you omit DHHC 10, and then you go straight on to DHHC 11, and then after that you go all the way on to DHHC 24. So even though there are whoops, missed off the C there. Okay, so that should be a C, and then 24. So even though there are 23 enzymes overall, the numbering system goes up to 24, and the reason is that you omit DHHC 10. There is no DHHC 10. Now, these are the names for the enzymes. These are not the names for the genes. If you're talking about the genes which code for these enzymes, the genes all have a Z in front of them. So the gene which codes for DHHC1 is ZDHHC1. Okay, the gene which then codes for DHHC9 is ZDHHC9. Okay, I won't write that because I haven't really got space. The gene which codes for DHHC11 is ZDHHC11. And then it goes all the way up to the gene which codes for DHHC24 is ZDHHC24. Okay, right, so these enzymes all catalyze the transferring of the palmitoyl group from the coenzyme A onto the cysteine residue, i.e. they palmitoylate proteins in the S fashion. So let me now discuss the structure of these proteins. Now, firstly, let me tell you the reason that they are called the DHHC enzyme family. Okay, so basically, in the structure of these enzymes, they all have a portion, okay, which is known as a serine, a cysteine-rich domain. Okay, I was thinking of the nuclear factor of activated T cells for a moment. Cysteine-rich domain, not the serine-rich region. Okay, so cysteine-rich domain is the structure that all of these have within them. Okay, and for short, the cysteine-rich domain is abbreviated to CRD. Now, the reason that these enzymes are called DHHC is that an important part of this cysteine-rich domain is a little strip that has the amino acid sequence DHHC. So in order to understand this, you need to know the single letter amino acid codes. Okay, so D is the single letter code for the amino acid aspartic acid. Okay, and in fact I might just draw these out for you. So let's actually draw this out. So we'll start over here with our aspartic acid. So here's the amino group of our aspartic acid. Here's the alpha carbon of our aspartic acid, the hydrogen of our alpha carbon, then the carboxylic acid group here, and the R group of aspartic acid. What you have is a methylene group and then a carboxylic acid group here. So this is the structure of aspartic acid. So you have an aspartic acid, then that's followed by a histidine, which is the amino acid which is represented by H. Okay, so this is a histidine amino acid. So let's put in histidine next. So here's the amino group, here's the alpha carbon with a hydrogen coming off it, and the R group of histidine is quite complicated. You have a methylene group, and then coming off that methylene group, you have an imidazole ring. Okay, so let me show this imidazole ring. So the imidazole ring is basically a 5-membered ring which consists of 3 carbon atoms and 2 nitrogen atoms. Okay, so here are 2 of the carbons, then you're going to have 2 nitrogens here, and then a carbon here. And as I say, well, as I've drawn rather, I haven't said it yet, but as I've drawn, you have a double bond there. Now you also have a double bond here, and this is the reason this ring is called and I've just put that exactly where I need to draw the next histidine. Great. I'll write it over here. So the reason this is called an imidazole ring is because the name for a double bond between a carbon and a nitrogen, as we have here, is an imide bond. Okay? So that's where the imide comes from in the imidazole ring. Now, of course, we just now need to add on enough hydrogens to saturate everything. So we'll have a hydrogen off this nitrogen, and this carbon also requires another hydrogen. Okay, now somehow I'm going to have to put another histidine in here. So here's the amino group. I'll try. Okay, so here's the alpha carbon. The R group is here. Now, can I incorporate that in somehow? Not really. I might put the ring going around that mistake. There's a solution. Okay, so the ring here, and this is an imidazole ring, so I'll put an I in the middle. Okay, so there now is the structure of the imidazole ring for this next histidine, so it's exactly the same as before. Okay, so then here is the carboxylic acid group that's bound to a peptide link, by a peptide link, to the next amino acid, which is cysteine. So here's the amino group, the alpha carbon again with a hydrogen coming off it, and the R group of cysteine, you have a methylene group, then with a thiol group coming off here. Okay, and then of course you'll have the carboxylic acid group, which will be involved in an amide link to the next amino acid along. So basically, in all of these enzymes, you have a cysteine-rich domain, okay, a CRD, and in this cysteine-rich domain, you have one of these little motifs, as it's called. It's called a DHHC motif, and it's part of the cysteine-rich domain. Okay, and this DHHC motif is conserved through all of these enzymes, and it's absolutely essential for their function, basically. In order for them to catalyze the transferring of this palmitoyl group from the coenzyme A onto the cysteine, you need this DHHC motif, basically. Okay, so let's now have a look at the structure of these enzymes, and they all have similar structure, although they don't have identical structure. So they all have one of these cysteine-rich domains, okay, and they're all transmembrane enzymes, so they have transmembrane alpha helices which anchor them in membranes. Okay, now they're all in membranes, okay, so they're all either in membranes in the endoplasmic reticulum, membranes in the Golgi apparatus, or membranes in the plasma, well, they could be in the plasma membrane. So, let me get another piece of paper and explain this. Okay, so, let's draw the different membranes that they can be in then. So, if this is our cell here, then another name for the cell membrane is just the plasma membrane. Okay, and the plasma membrane is one of the places that they can be. Okay, and they can also be in other membranes apart from the plasma membrane. However, here's the nucleus. Now, they're not generally found in the nuclear membrane, but I'll just put the nucleus there so that it looks like a cell. Okay, then you might have a structure here. Well, in fact, you will have a structure here, which we'll call the endoplasmic reticulum. Okay, so here is the endoplasmic reticulum, so I'll put ER, and then another membrane-bound organelle that you have within cells is the Golgi apparatus, and this is made up of a whole bunch of flattened membrane-bound structures all put together like so, and there's usually around 7 of these things stacked on top, but I've run out of space, so I've only drawn 4. Okay, now the ER and the Golgi are both very important in the production of proteins, basically, and the modifying of proteins so that they have their final structures. Okay, so Golgi apparatus. Right, so these enzymes can either be in the endoplasmic reticulum membrane, or they can be in the membrane of the Golgi or the plasma membrane. Now, basically, the way I'm going to draw this picture, I'm going to draw the phospholipid bilayer here, okay, and this side of the membrane will always represent either the lumen, okay, so if we're talking about the ER membrane, this will be the inside of the ER, or if we're talking about the Golgi membrane, this will be the inside of the Golgi, or if we're talking about the plasma membrane, we can't really call it a lumen anymore, the extracellular fluid. So if we're talking about the plasma membrane, this side will be the extracellular fluid, okay? And this side will always represent the cytoplasmic side. So if we're talking about the ER, then basically this side up here is the inside of the ER, and this side is the cytoplasm. If we're talking about the Golgi, it's the same. This side here is the inside of the Golgi, this side is the cytoplasm. If we're talking about the plasma membrane, this side is the outside of the plasma membrane, and this side is the inside, so the cytoplasmic side. Okay, right. So, depending on which which membrane they are in, will depend on whether this side represents the lumen or the extracellular fluid. Now, basically, the DHHC motif, which is within the cysteine-rich domain, will always be facing the cytoplasmic side of the membrane, whether we're talking about the endoplasmic reticulum membrane, the Golgi apparatus membrane, or the plasma membrane. Okay, so most of these DHHC family of enzymes have a structure that looks something like this. Okay, so they have 4 membrane spanning alpha helices, 1, 2, 3, 4. Okay, and this is their polypeptide structure spanning the membranes like so. Okay, and this would represent the amino terminus over here. So here's the amino terminus of the polypeptide, here's the carboxylic acid terminus, and this portion in the middle here is the cysteine-rich domain, the CRD, which has this DHHC motif somewhere within it. Okay, so most of the 23 DHHC enzymes have this structure. Okay, so let me now list out the DHHC enzymes which have this structure. So 1, 2, 3, 5, so 4 doesn't have this structure. Then 6 has this structure. DHHC7 has this structure, DHHC8 has this structure, DHHC9 has this structure, and it continues on. There is no 10. Then 11 and 12 both have this structure. 13 doesn't have this structure, 14 does have this structure, then it continues on. 15, 16, then 17 doesn't have this structure, 18 does, 19, 20, and 21 all have this structure, but 22, 23, and 24 do not have this structure. So most of them do have this structure, basically. So this is your best guess as to the structure of a DHHC enzyme. However, some of them do not have this structure. So for instance, DHHC4 does not have this structure, DHHC13 doesn't have this structure, and DHHC17 doesn't have this structure, and then 22, 23, and 24 don't have this structure. So, let's now look at the freaks, basically. Let's look at the ones that don't obey this structure. So, we'll turn our attention now to the DHHC4 enzyme, okay, which is on its own, basically. There's no other DHHC enzyme which has a structure similar to DHHC4. So basically it's almost the same. So let's draw the one that we know, the main one here again. Okay, so here's the cysteine-rich domain here. This is the CRD. However, what you're going to do is you're going to add on another membrane spanning alpha helix there, and the amino terminus will then be on either the luminal side of the membrane or the extracellular side of the membrane. And of course the carboxylic acid terminus will still be here. So this is the structure of DHHC4. Now let's turn our attention to 13, 17 and 23. So 13, 17 and 23 all have a very similar structure. Okay, so they all can be put together as having the same membrane spanning topology. Okay, so 4 was the first freak. Now we're turning our attention to 13, and it has the same membrane spanning topology as 17 and 23. And then we'll deal with 22 and 24 finally. Okay, so 13, 17, and 23, their membrane-spanning topology looks like this. They actually have 4 membrane-spanning alpha helices before they then have the cysteine-rich domain here, and then a final 2 membrane-spanning alpha helices here. Okay, so here's the amino terminus, and there's the carboxylic acid terminus. So this is the membrane-spanning topology of 13, 17, and 23. But again, note that the cysteine-rich domain is always on the cytoplasmic side of the membrane, whether we are talking about the plasma membrane, the endoplasmic reticulum membrane, or the Golgi apparatus membrane. Okay, now let's have a look at DHHC22. Okay, so again, DHHC22 sits on its own as a lone DHHC enzyme. Like DHHC4, it has no other enzyme that's similar structure to it. So it only has 2 membrane-spanning alpha helices. Okay, so it's now used up all its membrane-spanning alpha helices. It then has the cysteine-rich domain here, the CRD, before it finally ends with its carboxy terminus here. Okay, and then here is the amino terminus of this protein. And then finally, let's discuss the structure of DHHC24. Okay, so the structure of DHHC24, it has 5 membrane-spanning alpha helices, like DHHC4, but the additional one is on the opposite side to DHHC4. So it will have 2 here, so the amino terminus will be on the cytoplasmic side. Then it will have the cysteine-rich domain here, so here is the CRD, and then it will have the 3 membrane-spanning alpha helices like so, with its carboxylic acid group either facing into the lumen or facing into the extracellular fluid, depending on which membrane we're talking about. And this is the membrane-spanning topology of DHHC24. So, the DHHC enzymes all have a similar structure. The important thing to understand is that this cysteine-rich domain is essential for their palmitoylation function. Okay, and within that cysteine-rich domain there is this conserved motif, this DHHC motif, which is aspartic acid followed by 2 histidines and then followed by a cysteine residue, which is essential for the palmitoylation function. Okay, right, so we'll continue this discussion in the next video, where we'll look at which membranes all 23 are in, basically. So which ones are in the ER membrane, which are in the Golgi, and which are in the plasma membrane.